So today we're going to be talking about prokaryotic form and function, which covers chapter 3. Now this is a really critical lecture because it will kind of catapult into the rest of everything that we'll be talking about this semester. So we're going to start small and learn about what's inside bacterial cells, and then we'll build from there. So it's really important that you understand the different structures and their functions of what we're going to discuss today. So first, let's set the stage. Basically, we've got two types of cells. We've got prokaryotic cells and we've got eukaryotic cells. Today, we're going to talk all about prokaryotes. Next time, we'll talk all about eukaryotes. And how these two differ there are three main points of how they're different. One is the way their DNA is packaged. Meaning prokaryotes don't have a nucleus. They also lack histones, which are part of DNA in eukaryotic organisms. Another thing, the makeup of their cell wall. Most eukaryotic cells don't have a cell wall. The ones that do not contain what we call peptidoglycan. It is a very unique chemical to prokaryotic cell walls. And then there's also differences in their internal structure. Prokaryotes lack what we call membrane-bound organelles. These are little organs that perform specific functions in eukaryotic cells. So only eukaryotic cells are going to have those organelles. Prokaryotic cells are going to be much more simple, and they will lack those membrane-bound organelles. So let's just jump right into the parts of the prokaryotic cell. So I have it broken into three different groups. We've got parts that are in all bacteria, parts that are in most bacteria, and parts that are in some bacteria. So in all bacteria, we're going to find a cell membrane, cytoplasm, ribosomes, a cytoskeleton, and one or a few chromosomes. In most bacteria, we're going to find a cell wall and a surface coating called a glycocalyx, and we can also call it a capsule. We'll talk a little bit more about that in a moment. And then in certain bacteria, in some bacteria, you'll find flagella, pili, and fimbriae. You can find an outer membrane. We can find plasmids, we can find inclusions, and we can find endospores. And something I want to point out, it says at the top of these little tables, in all bacteria, most bacteria, some bacteria, and at the top I have parts of the prokaryotic cell. So we're going to use the terms prokaryotic and bacteria interchangeably. The only other prokaryotes out there are Archaea, which we talked about last time. Archaea we're not going to discuss in this class at all, and so for our purposes, prokaryotes equal bacteria. So if you see prokaryotic cells, for the rest of this lecture, we're talking about bacteria specifically. So before we start talking about the details of each of the parts of the prokaryotic cell, I wanted to put this figure in here to kind of get you looking at everything you need to know about the prokaryotic cell. So you'll need to know each of the prokaryotic structures listed, their functions, and their relative locations in the cell. And this figure can help you visualize that. Also, check out the flowchart on page 62 to help with the general organization of the cell, about what is where and basically what the little parts do. And again, on the subsequent slides, we'll be talking about what each of these parts does, but this slide can kind of help you organize your studying so you can see all the parts at one time. So prokaryotic cells tend to have a characteristic shape, and we call that characteristic shape morphology. So since bacteria or prokaryotes are single-celled, we call this cell morphology because each organism is one cell and that cell has a particular type of shape. And so I have the different shapes listed here. So we can have a coccus, which is a spherical or a ball-shaped cell. It's at the top of the photos there. We can have a bacillus. The plural of that is bacilli. So if we have more than one bacillus, it's called bacilli. And those are rod-shaped or rectangular. And so they can be really blocky, just like a regular rectangle would look like, or they can have tapered or rounded ends. We can also see something called a cocobacillus, which is a short and plump bacillus. It's basically the mix between a coccus and a bacillus. Then we also have spiral-shaped prokaryotes, and there are three different types of spirals. We have the vibrios, which are curved rods. They're an incomplete spiral. They're more of a C shape. You can see they're the third photo down on the list there. We also have sporulum, and these are spiral-shaped organisms that have a very rigid spiral shape. So we call them helical. But the key thing is that they're rigid. They stay in that specific shape. A spirochete is a spiral-shaped organism that is flexible. It has the appearance like a spring or like a slinky. It's much more flexible in terms of what it looks like. And then finally we have branching filaments. That's our last photo there. And basically there's a rod shape that's centrally located, and then we have branches coming off of that basic rod shape. So prokaryotic cells not only have a specific morphology or shape, they also have a specific arrangement, meaning when there's more than one cell, how do they congregate together? And so we have cocci arrangements, which is what we have for the coccus shape, and we've got bacilli arrangements. So cocci arrangements, we can see cocci in diplococci, which are pairs of cocci. We can see them as tetrads, which are groups of 4. We can see them as cubical packets of 8 to 64 cells, and we call those Sarciniae. We can see them as clusters, which we can refer to as staphylococci and micrococci, or we can have chains, which are also known as streptococci. Now bacillus, we see in different types of arrangements. We see them in less types of arrangements because they only have one axis which they can divide on, and so they're much more limited at how they congregate. So we can have a diplobacillus or a diplobacilli, which are pairs, chains, which are known as streptobacilli, and then we also have what is called the palisades, which are rows oriented side by side, and I have a photo there of what the palisades arrangement looks like. So we've talked about general prokaryotic morphology and arrangement. So now let's talk about the actual structures associated with the cell itself. So we've got external structures that are outside of the cell wall, which we'll talk about the cell wall in a few slides. It's a very important prokaryotic feature. So we'll talk about the external structures and then we'll move towards the inside. And so outside of the cell wall, we see appendages for movement. So bacteria don't just stay in one place, they move around, and some bacteria have these appendages for movement. And those appendages include flagella and axial filaments. Now flagella, it's basically like a little boat propeller. It's a microbial propeller that has 3 main parts: the filament, the hook, and the basal body. And this little propeller is what moves a bacterium towards or away from something good or bad. And flagella can have polar arrangement, meaning it's only at the extremes of the cells, and you can see that to the right of the words here, or a peritrichous arrangement, which means it's just randomly found all over, and that's to the right, far right of the slide. Now, I mentioned it allows an organism to move towards something good or away from something bad, and we call that chemotaxis. So it's moving in response to a chemical signal. And the way the movement actually works, it occurs in this run and tumble manner, to where they go along at a straight pace for a little while, and then they kind of tumble, and then they move into a different direction. And you can see that in the box, that figure is in the box to the right of the run and tumble manner. Now, flagella can be found in a variety of bacteria, but axial filaments are an appendage for movement that's only found in the group of known as spirochetes. So remember, those are going to be our spiral-shaped organisms that we just talked about on the last couple of slides. So we just talked about appendages for movement. Those are some external structures outside of the cell wall. We also have outside of the cell wall appendages for attachment and mating, and those are called fimbriae and pillae. Now fimbriae are short bristle-like fibers, and they're all over the surface of the cell, and they help the bacteria stick to surfaces or adhere or attach. Those are all words for the same thing. And these fimbriae help bacteria form biofilms on inanimate objects, and inside a human host they help them attach to a host cell, which makes it great because it anchors them down and it kind of lets them grow and be in this nice happy place, which leads to colonization and disease. So I said it's great for the microbe, right? Not for us. So fimbriae play a large role in that as well. Now pillae are external structures used for mating. They are these long rigid tubes that are used in the process of conjugation, which we'll get to in a few weeks. But keep in mind, a fimbriae is going to be these short bristle-like hairs all over the surface of the cell, helps with attachment. Pillae are going to be long rigid tubes. Typically you're only going to see 1 or 2 on a bacterium, and those are used for mating or conjugation. So what else is outside of the cell wall? We have a surface coating known as the glycocalyx, and on a few slides back I mentioned that a glycocalyx is something that most bacteria have. So this is not something that's in all bacteria, but most bacteria are going to have this surface coating. And what it is, it's this sugary coating on the outside of the cell wall that's found in many bacteria. And it can help with the formation of biofilms. And there are two types depending on what they look like or what they're composed of. We can have a slime layer or we can have a capsule. So slime layers and capsules are two types of glycocalyxes. So a capsule is a type of glycocalyx. A slime layer is a type of glycocalyx. So a slime layer is this thin coating that is loosely attached to the cell wall. It's kind of unorganized, it's just kind of hanging out. Now a capsule though is a thick, dense coating that is firmly attached to the cell wall. It's highly organized and it's commonly formed by pathogenic bacteria. It not only helps them form biofilms, but it provides protection from the immune system. It's kind of like an invisibility cloak. Capsules keep a bacteria from being phagocytized, which is our body's way of clearing out foreign invaders and garbage inside of our body. So phagocytosis is an important process we'll talk about later this semester. But basically the capsule keeps the bacterium from being seen by the immune system and from being gobbled up through phagocytosis. So slime layers, capsules, two types of glycocalyxes. Now macroscopically speaking, a capsule can have an effect on what a colony looks like. So I want you to take a look at this figure to the right of the page. The top part of the plate is a non-encapsulated bacterium, and you can see it's grown, it just looks white, plain. On the bottom though is the same bacterium, but it is encapsulated, meaning it has a capsule. And you can see it does look quite a bit more slimy, or what we call mucoid, because of the presence of that capsule. And so just wanted you to note that the non-encapsulated on the top and the encapsulated on the bottom even physically look different because of the presence of that capsule. And they're also going to behave differently because of the benefits that that capsule offers. So finally, we've been talking about what's outside of the cell wall, but what is the cell wall? So the cell wall is a really important bacterial structure that helps determine the shape of the bacterium. It helps give the bacteria structural support, and it helps prevent the rupture of the cell due to osmotic pressure, which is basically if there is salts and sugars in the environment, it helps keep the cell from bursting because there's too much or too little around. So it helps keep the cell balanced as far as osmosis goes. Bacterial cell walls have this really unique component that I mentioned earlier in this lecture. It's called peptidoglycan. And this peptidoglycan is basically these long sugar chains that are cross-linked to each other by peptide fragments. And it allows the bacterial cell wall to be very strong, but also flexible. Okay, so we've got these two major types of cell walls that we see in bacteria. We've got gram-positive cell walls and we've got gram-negative cell walls, and they look very different from each other. So gram-positive cell walls, they have this really thick peptidoglycan layer. Now don't forget, we just said peptidoglycan is that unique component of bacterial cell walls, and it's these long sugar chains connected by peptides, little proteins. So it's got this really thick peptidoglycan layer, and then it's got these teichoic acids and lipoteichoic acids running throughout. So you can see in the figure below, on the left-hand side, we've got this gram-positive cell wall. And all of that peptidoglycan is that really thick purple layer. And those little orange chains running throughout, those are going to be our teichoic and lipoteichoic acids running through. Now, gram-negative cell walls look quite different. You can see just from the photo. They just have a very thin layer of peptidoglycan, which you can see represented by that really kind of magenta color that's running through the center of that figure. Now, on top of that thin layer of peptidoglycan, we have what is called the outer membrane, and that's what those little that little layer on the top looks like, and it is called the outer membrane. And the outer membrane contains what we call LPS, which is known also as endotoxin, which is an important virulence factor. It helps with producing disease, producing symptoms of shock, things like that. And we'll talk about that more this semester. But it's really important that you understand these key differences between the two cell walls, the main structures. It's really important to point out that at the base of both of these figures, for both the gram-positive and the gram-negative cell wall, we have what is called the cell membrane. And we'll talk more about what the cell membrane does in a few slides. But those like orange balls with the little tails that are forming that bilayer at the bottom, That's the cell membrane. That is not part of the cell wall. That's what the cell wall rests on. So it's really important that you understand that the cell membrane isn't part of the cell wall. What's above it, that's what we consider to be the gram-positive cell wall and the gram-negative cell wall. So in 1884, this guy Hans Christian Gram, where this name of the stain comes from, decided to try out this technique, and it was the sequential application of these different dyes. And that's what is known as the gram stain method now. And as he did this, he realized that it did distinguish between two types of organisms, and that is due to the fact that they have different types of cell walls. And so it's really important to understand what's happening at each stage of the gram staining process, both what the cell looks like microscopically and chemically what's happening in the cell wall. And this is a nice figure from your book that helps you walk through that process. And so we're going to go through that right now. So gram staining is 4 main steps. It's the addition of crystal violet, the addition of grams iodine, the addition of alcohol, and then the addition of safranin. So let's start at the beginning, crystal violet. Crystal violet is what we call the primary stain. It's the first stain that we add. Like the name implies, it is purple. So when we add it to both the gram-positive and the gram-negative cell wall, both of them start to look purple. And that's because the gram-negative and gram-positive cell walls take the dye up. They take those crystal violet molecules up within the cell wall and keep them there. Now, when the grams iodine is added, this is what we call the mordant, and it basically complexes with those crystal violet molecules and makes them really large to where they cannot escape from the cell wall. And so that dye complexes in the cell wall and it keeps that crystal violet there. And so it's only from the iodine complexing with the crystal violet that makes it large enough to get trapped inside there. Now next we add alcohol. And what the alcohol is going to do is dissolve that outer membrane in a gram-negative wall. And because that outer membrane is weakened, that wall, remember there's only that thin layer of peptidoglycan that has the crystal violet trapped inside, it's exposed, and so that dye just kind of leaks out. Okay, but remember the gram-positive cell wall has all those layers of peptidoglycan. Remember the slide before is that really thick layer, that really thick purple layer? Well, all of those molecules are still trapped inside there, and that alcohol isn't really breaking down the peptidoglycan enough for the molecules to escape. So the crystal violet remains in the cell wall of the gram-positive organism. It escapes from the cell wall of the gram-negative organism because the outer membrane has been dissolved by that alcohol. The final step is going to be safranin, which is a red dye. It's also kind of pinkish. It is known as the secondary stain because it's the second color we're going to use in this process. And basically both types of cell walls take up the dye. So the gram-negative cell wall takes it up, the gram-positive cell wall takes it up. However, there's so much purple stuck in the gram-positive cell wall that it masks that red safranin. And so in the case of the gram-negative, it's colorless from the alcohol, so it takes up that red dye and becomes reddish or pink. And so at the end, you have this purple gram-positive cell and pink gram-negative cell, pink or red. So I mentioned the 2 main types of cell walls are gram-positive and gram-negative. So just because we have 2 main types doesn't mean we don't have other strange things happening in the microbial world. We also have what we call non-typical cell walls. So acid-fast bacteria are some of those non-typical cell walls. We don't really consider them to be gram-positive or gram-negative per se. We consider them to be acid-fast. And those include the bacterial genera Mycobacteria and Nocardia. And these acid-fast bacteria contain what are known as mycolic acids. They're very waxy, and so they're very resistant to dyes and other things. And so these acid-fast bacteria are considered modified gram-positives, but they're technically their own kind of cell wall. Archaea, remember I mentioned earlier, we talked about archaea, that they are prokaryotes, but they're classified in a separate domain from bacteria. So we've got 3 domains of life. We've got bacteria, eukarya, and archaea. Now archaea do not have peptidoglycan in their cell wall. So that is something important to note. And then we also have what are known as the cell wall deficient bacteria. These guys have no cell wall at all. Their cytoplasmic membrane, which we'll talk about in a minute, is stabilized by sterols, and that kind of helps keep those bacteria resistant to lysis. Remember, the cell wall, that's one of the important things, is it helps keep the organism resistant to lysis. So if you don't have a cell wall, what will happen? And so in that case, they've evolved special mechanisms to get around that. So under the cell wall is what is known as the cytoplasmic membrane. So we've had our structures external to the cell wall, we've talked about the cell wall, and now we're going under the cell wall. And the first thing immediately under the cell wall is the cytoplasmic membrane. And this is a phospholipid bilayer that has embedded proteins. Now, phospholipids, these are going to be a type of lipid, a type of fat, and that we'll see compose the majority of the membrane. And so 30 to 40% of the membrane mass are these phospholipids, and that's going to be the kind of main structure. And then embedded within this main structure, we've got proteins. And so the proteins take up more mass, they're heavier, but the phospholipids make up the bulk of the structure itself. Now, the function of the cytoplasmic membrane, also known as the cell membrane, also known as the plasma membrane, the function of this structure is to regulate transport of molecules. That's its primary function. It is selectively permeable for nutrients and molecules to pass in and out of. So basically, we call this the bouncer of the cell because it is saying, hey, you can come in, now you need to go out. So it's just regulating what's coming in and out of the cell, and that's important because you don't want everyone coming in at one time, you don't want everyone leaving at one time. It also deals with some energy reactions and metabolic activities. That's more of a side function. It contains enzymes for some types of metabolic processes that we'll talk about later on this semester, but primarily its function is to regulate transport. So within the cytoplasmic membrane is what we call the cytoplasm. So the cytoplasmic membrane completely surrounds this gelatinous solution known as the cytoplasm. And it's primarily water, but within the water matrix we've got proteins and salts and carbohydrates, and basically it is the site of action for all types of things in the cells. It's the party place. It's where all types of things are going on. So anything that's bound by the cytoplasmic membrane is known as the cytoplasm. It's the matrix that suspends the other little pieces of the cell that are within that cytoplasmic membrane. And we'll talk about what those pieces might be in just a moment. So here are the pieces. I said there are little pieces of things in the cell and suspended in the cytoplasm, and these three are some examples of what can be found inside the cytoplasm. So we've got the nucleoid, we've got the ribosomes, and we've got inclusions and granules. So the nucleoid is the area where the bacterial chromosome is found. So it's going to be the genetic material that will tell the cell what to do. It is not enclosed by a membrane, so it's not considered to be a true nucleus like we will see next time eukaryotes have. So eukaryotes have these true nuclei, but prokaryotic cells only have what is known as the nucleoid. The nucleoid area can also contain plasmids, which are optional pieces of DNA that can give a bacterium protective traits, like antibiotic resistance, for example. The ribosomes, which is what we see in the figure to the right of the slide, those are the sites of protein synthesis. So there's lots of ribosomes that are suspended in the cytoplasm. The size of a prokaryotic ribosome is different than that of a eukaryotic ribosome. Prokaryotic ribosome is called 70S, and it has a large and small subunit. A eukaryotic ribosome also has a large and small subunit, but it's known as an 80S ribosome. Another thing we can see suspended inside the cytoplasm are inclusions or granules. We can kind of think of these guys as storage closets for nutrients, for gases, or for any other types of molecules that serve a purpose in the cell. So they're just like a little storage area that the cell can use for later on. So some bacteria have the really cool superpower of being able to form what we call endospores. And it's an optional survival pod. It is a way for bacteria to survive times when they don't have what they need in the environment to survive. They don't have the right temperature, the right pH, there's not enough nutrients, there's not enough water. And so at that stage, certain bacteria, certain species of bacteria can form these endospores, which are dormant bodies, meaning they're going to kind of go to sleep. and that's formed in response to hostile conditions, and it helps the organism survive those hostile conditions until favorable conditions are present. And so the species that are most, or the genera that are most associated with endospores are Bacillus and Clostridium. And remember that this is a dormant body, so it's just a state of rest. They're not reproducing, they're not really doing anything, they're just kind of vegging out. They're just kind of hanging out, waiting for the coast to be clear. Now, the formation of spores is known as sporulation, so definitely circle that. That's a good word. And then the return of the spore to the vegetative state, or the normal bacterium, so a normal bacterium is known as a vegetative bacterium. That is what we call germination. So spores to normal bacterium, that's called germination. Bacterium to spores, that's called sporulation. Now, germination happens when favorable conditions come up, and it can happen really quickly. Now, the reason we bring up endospores is because they are very important in terms of our understanding of bacteria and how they respond to different conditions. Endospores can help them survive much harsher conditions than normal bacteria can survive. And so this is important when we think about applications of microbial growth control, trying to kill bacteria with heat, with disinfectants. Endospores are extraordinarily resistant, so it's really important for us to understand that there are these survival pods out there, and we need to know about that in terms of treatment and disinfection and other types of microbial growth control. Okay, so let's look at the process of sporulation a little more closely. Now remember, sporulation is the process of a vegetative cell turning into a spore. And so first you have this vegetative cell beginning to be depleted of nutrients, and so that's indicative of the fact that the environmental conditions are not getting so great, so we want to maybe think about forming the spore. And so then, of course, the chromosome is duplicated and separated, and then the cell forms this little wall. It's septated. That's what that means. It forms this little wall that forms a sporangium and a forespore. Now, the sporangium is going to be more like the big part of the cell. The forespore is going to be the little part that will become the actual endospore. And so the sporangium engulfs the forespore for further development, and then it begins to synthesize spore layers around the forespore. So the forespore is going to be this early endospore, and so more layers are deposited, and finally the endospore is mature. But you can see on step 7, this is something that's still inside the sporangium, so it's still within something else, so it's not quite finished. So finally the spore is released, the sporangium is lost, and so you just have this endospore by itself. Now, when it's time for favorable conditions coming around again, then we have germination. And so the spore swells and it releases the vegetative cell, so it goes back to being a normal cell. So that's basically the cycle of turning into a spore and turning back into a vegetative cell. So bacteria are not the only prokaryotes on the planet. Sometimes we forget about that. We also have archaea. So remember, those are a different domain than bacteria, but they are both prokaryotic types of cells. So in terms of bacteria versus archaea, what do they share and how are they different? So they both have a cell wall, but archaea do not have peptidoglycan cell walls. So bacteria are going to have that peptidoglycan cell wall, and that's the only place we see that in the realm of biology. So you need to make sure you understand when you see peptidoglycan, we're talking about bacteria. Archaea share more characteristics with eukaryotic cells than bacteria do. So for example, archaea have more close rRNA sequences, they have more similar protein synthesis, they have more similar ribosomal structure to eukaryotes than bacteria do. So archaea seem to have more similarities with eukaryotic cells than bacterial cells have with eukaryotic cells. A key characteristic of archaea is that they are extreme. They are often found in extreme hot or cold temperatures or at very high osmotic pressures. And so this is unusual because these guys are living outside the range of what normal organisms can tolerate. So we call them extremophiles, that type of thing. And that's normally those extremophiles are going to be archaea. So remember last time we talked about taxonomy and classification, we talked about kingdom, phylum, that type of thing. So bacteria are classified just like other organisms, just like we spoke about last time. from kingdom down to species. However, they're different than other organisms in the fact that they can have divisions and classifications below the species level. And so we use words like subspecies and strain and type to designate bacteria that belong to the same species, but they have different characteristics. So they are the same species, but there's something a little bit different about them, either physically or chemically or biologically, that causes them to be called a subspecies or a strain or a type. And so serotype is a term given to members of the same species that stimulate a distinct pattern of immune response in the host. For example, Vibrio cholerae, it's an agent that causes cholera worldwide. You might not know about cholera yet, but you will find out more about it this semester. This organism has multiple serotypes, meaning that this one particular species, there are several different types of the organism that cause different immune responses in the host. And so we have two really famous ones, V. cholerae 01 and V. cholerae 0139, and these are very famous serotypes because they have specific toxins that they produce and as part of the effort against our immune response. And so serotypes are going to be a type of classification below the species level. So that's important that we discuss this terminology for classifications of prokaryotes because it's a little different than what you might have seen in other realms of biology. All right, so let's apply what we've been talking about so far with a focus on disease. And we're going to focus on the disease tetanus, which is commonly called lockjaw. And it's caused by the bacterial agent Clostridium tetani. And you might remember from a few slides back, I mentioned the genus Clostridium as one of those genera that is capable of producing endospores. And that's why we're talking about this guy, because endospores help Clostridium tetani survive in the environment. So it can withstand all types of things that most bacteria would not be able to. And so this agent doesn't need oxygen, so it grows great in puncture wounds. And that's usually how this goes about. You are outside, you step on something, you get a puncture wound. This endospore was helping the agent survive in the soil, and now that's introduced into the wound, which is a nice anaerobic area. And so the spores usually germinate in the wound, meaning now they found favorable conditions, and so they go back to being a vegetative bacterium, and so then the organism grows, and in the process it produces a toxin known as tetanospasmin. And this tetanospasmin produces the very characteristic paralytic symptoms of tetanus, including causing lockjaw. Now, vaccine and the boosters prevent the disease, which is why, if you ever step on something outside, the first thing they do to you in the ER is they ask you, have you had your tetanus booster lately? And they'll try to give you one. And so that's a great way of preventing the disease. And there are 4 different forms. We've got neonatal, local, and cephalic. Those are super rare. But then we also have the generalized form, which is the most common form. It's what we'll see most often in the hospitals. So keep in mind, endospores, remember we said they're very hardy, they're very resistant, and in the case of Clostridium tetani, it's what helps it survive until it gets into a new host. So this concludes our talk on prokaryotic cell structure and function. Be sure to bring any questions you have to your instructor.